Hello, my name is Elena Georgiou. I am postdoctoral fellow in the Neuroscience Department, directed by Professor Kleopas Kleopa at the Cyprus Institute of Neurology and Genetics in Nicosia, Cyprus. Our institute is a leading not-profit national medical and research center that has been active in the areas of biomedical research, specialized services for patients with neurological and genetic disorder, and postgraduate education for over 3 decades in Cyprus and the wider region. My research on gene therapy for PMLD has been ongoing since 2013 when I joined the research team under the supervision of Dr. Kleopa. Pelizaeus Merzbacher-like disease type 1 or hypomyelinating leukodystrophy type 2 is an ultra-rare inherited neurological disorder characterized by progressive degeneration of the white matter of the brain and spinal cord starting in early childhood. The disease is caused by autosomal recessive gene JC2 gene mutation that lead to loss of the function of the gap junction protein connexin 47. Connexin 47 is expressed specifically in oligodendrocytes, which form the myelin sheath throughout the central nervous system, and it is necessary for the communication between cells and preservation of their integrity. In our research lab, we previously demonstrated that an adeno-associated viral vector delivering the healthy human gene JC2 gene injected directly into the brain leads to a significant therapeutic response in a mouse model of PMLD1. In recent years, we have further optimized our therapeutic approach by testing more relevant viral vectors and injection methods that are currently used in clinical practice for other disorders. We identified AAV9 as the most promising vector and intravenous injection as the most effective in delivering the vector to the brain, at least in young animals. This approach has confirmed the therapeutic benefit in the disease model. Thus, we just started a dose escalation and toxicity study to clarify the minimal effective dose. This will help to find the optimal dose in order to improve the delivery of the vector into the brain and spinal cord, which can be adequately scaled up in both young and older subjects with a mature blood-brain barrier. This work will provide further proof of concept for the applicability of our gene therapy, especially in young children with PMLD1, a similar approach currently in clinical trials for another leukodystrophy, Canavan disease. We are getting close, but in order to make progress and successfully complete the remaining steps towards the clinical application of an effective treatment for PMLD1 patients, we need to mobilize industry support for the next steps, as well as all patients and their families worldwide to create a critical mass for pursuing this goal. We would like to establish a global consortium with all patients suffering from PMLD, all expert clinicians and researchers. The aim of the consortium will be to exchange knowledge, create a unified patients registry, and start a natural history study, develop relevant biomarkers, and lobby for therapy development. Every family living with a PMLD1 is important and should be involved in our efforts. If you are a family with a child or children with Pelizaeus Merzbacher-like disease type 1, you may communicate with PMLD support group in Facebook and should also register at the Children's Hospital of Philadelphia, which has started an initiative to collect robust natural history data using a Children's Hospital-based patient registry as a platform. This registry, funded by Dr. Adeline van der Veer, known as the Myelin Disorders Biorepository Project and Global Leukodystrophy Initiative Clinical Trial Network, has enrolled over 2,000 individuals with a variety of leukodystrophy and plays a vital role in the design of natural history studies. European patients may also register with one of the centers participating in the European Reference Network for Rare Neurological Disorders that has a working group on leukodystrophies. The natural history study will be a crucial step for better understanding the etiology, range of manifestations, and progression of PMLD1, as well as for attracting industry support to bring therapeutics to the clinic. Finally, researchers in Rome and Milan work on the development of biomarkers of the disease, and based on patient samples and participation in their study will facilitate future clinical testing of treatments. Please help us to make the most of the exciting research developments around PMLD1 and to create a global collaboration for advancing care and cure for those living with PMLD1.